All right, we're going to talk about our prokaryotes. We've done this. Building a cell. Cells are mechanical. Now we're going to talk about prokaryotes and then how we get into eukaryotes. So prokaryote, let's define that little term for ourselves now. Let's do our prokaryotes. We'll just do them in this little black color. Prokaryote. What does that mean? And then we'll also write up here eukaryote. Eukaryote. Okay, well, this means it's like proto. It's like prototype. Prototype. Think of it like that. Prototype means new, kind of early, or a fancy word would be nascent. Those all kind of mean the same thing. Prokaryote, what does karyote mean? What does this mean? Karyote means nucleus. So it's a new, kind of early, nascent, semi-nucleus. It's kind of a nucleus. You can really think of it as this one. It's probably the best one. It's kind of a nucleus. What does this mean? This means true. And this means, again, nucleus. Nucleus. So a prokaryote has kind of a nucleus. All right, well, what's a prokaryote? Let's get into it. We're going to draw them in black here, nice and easy. All right, prokaryotes, something like this. And you can just think of them as bacteria. So it's also archaea, but we can just think of them as bacteria. That's all I care about. They're bacteria. A prokaryote is a bacteria. They don't have a nucleus. They have DNA like we do. And their DNA is a big circular molecule. I'm not going to draw both strands here. This is just one strand. It's all in the center. I haven't completed the circle, but now I'm going to, boom, like that. So they have circular DNA in the center. It's not a nucleus because there's no membrane around it. It's called a nucleoid. Nucleoid. Called a nucleoid. Okay? It's pretty nifty of them to have that. They got a nucleoid. Okay, so that's just one way that they share they have genetic information stored. Another way that's pretty nifty is they have tiny circular DNA. Pretty cool. They also have tiny circular DNA that can store a gene or two. That are important for survival. These are called plasmids. All right, we'll come back to these. So they have these tiny little genes. So like, for example, something that they might store, it's important for survival. Something like antibiotic resistance. So something like resistance against penicillin. Resistance against penicillin. Don't know how to spell penicillin. There we go. Resistance. Perfect. Resistance against penicillin. All right, cool. What else I want to talk about? Nucleoid. Okay, so they will also have structures that come off of them. For example, that will allow them to swim, kind of like a sperm. Let me draw this a little thicker, actually. It's called a flagella. Flagella allows them to swim. So this thing will actually have a big old base like this that kind of sits in the membrane, like so. And then this base right here has a lot of cool machinery involved in it, and it will spin. The base will spin, causing the flagellum to whip around, and that's how they swim. Flagellum. Flagellum. All right, that's pretty cool. Another thing that I want to talk about is having to do with these plasmids. All right, these plasmids. So sometimes bacteria, let's get a little fancy here. Well, let's draw another bacteria over here and do something pretty special. We got another little bacteria over here. A little flat on the bottom because the thing was in the way, but it's okay. So this bacteria here will actually produce a big, long bridge between these two. It's a very thin little projection, like so. Oops. Bang. Okay, this thing is called a conjugation pili. Pretty cool. Conjugation pili. Pretty nifty. And this bacteria would, of course, have its nucleoid, like so. But let's say it doesn't have these plasmids that this bacteria does. And let's say maybe there's some penicillin around here. So this bacteria can actually transfer, it can make copies of these plasmids, transfer them through this conjugation pili to this other bacteria. Conjugation pili allows bacteria to exchange genetic information. Pretty freaking cool. Exchanging of genetic information, another word for that is bacteria sex. Exchange genetic information. Bacteria can have sex with these conjugation pili. Conjugal visits at prison. I mean, it's right there in the word. Yeah, who doesn't like to Netflix and conjugate? Bacteria do. They love it. What's the new show? What's the haps? That's pretty nifty. So, you know, this bacteria can like help out its buddies. Dang, like a bird just hit my window. You know, help them survive. So they'll have very important genes on these little plasmids. And they can exchange this genetic material through these conjugation pili. Okay. All right. Let's zoom in again here. I'm not going to draw all of this. But if we look at these bacteria, they will actually have, there's actually a couple different types of bacteria. And some of them will have membranes, they'll have two membranes. So they'll have that phospholipid bilayer, and then they'll have another one, like so. Then they have a little space in between there, and that space is full of a polysaccharide, actually, a carbohydrate. So this is one type of bacteria. This is how E. coli is organized. I'm trying to think how I actually want to draw this. Let's draw this differently. Let's do a little. I gotta I'm thinking about your guys' notes, and I'm going to put this in a way that you can actually take notes on this. So gram-positive and gram-negative bacteria. Graham was a microbiologist who figured out this stain. So there's gram-positive bacteria and gram-negative. And so if we look at the membrane of these bacteria, they look slightly different. So we just look at just the membranes of a gram-positive. They will have a single membrane, and then they will have a huge peptidoglycan layer. I want that to be filled in. So they will have cell membrane, and then peptidoglycan layer. So peptidoglycan, it is a peptide and a carbohydrate. That's what that is. Okay, so those are gram-positive. You add a crystal dye to them, it sticks to the gram, or sticks to the peptidoglycan layer, so they will look purple. These will look purple when dyed. Not that kind of dye, dyed. It will look purple when dyed. And we'll actually do this, stain it. We don't actually do that in this lab, but we'll see some of these under the microscope that have been stained for us, okay? Gram-negative, very similar, except they have two membranes. So they have an inner and an outer membrane. So we've got, whoops, ooh, I got kind of a bellyache. Got an outer membrane, and we've got an inner membrane. And we've got a little thin layer of peptidoglycan in between them. So we've got an outer membrane, and an inner membrane. Then we have our peptidoglycan layer in between. And peptidoglycan is a peptide and a sugar. Let me do that up here for you. This is a protein and a sugar. Oh, macromolecules. You mix them together, you get peptidoglycan. These gram-negative bacteria will look pink. Pink when dyed. We wash away the purple dye, so they will look pink. Examples of these will be, no, not that kind of dye. Oh, I feel like my power might go out here in a second. That would suck. When dyed. Gram-positive, an example of this would be Staphylococcus aureus. This is a bacteria that lives all over your skin. Staph aureus, but you can get staph infections, and that's what they're talking about on your skin, is when your Staph aureus gets out of control. Staphylococcus aureus. And an example of gram-negative bacteria would be our fan favorite, Escherichia coli. Escherichia, so close. Escherichia. E. coli. That's E. coli. Sure you've heard of that guy before. E. coli. This would be Staph aureus. Okay, that's gram-positive, gram-negative. All right. So with the gram-negative bacteria, I'm going to go back up here. They do their energy production right here. So they make their ATP. So they got like little proteins embedded in their membranes. And they make all their energy right up in here. So they make their energy up in the space between the membranes. Pretty nifty how they do that. Pretty cool, I gotta say. the space allows them to do it. That space is called the periplasm. I don't necessarily care that you know the name of that space. All right, so bacteria are like little energy producers, right? They're like tiny little energy producers. Okay, so let's get to this question now. That's kind of all I want you to know about prokaryotes. How did this transition occur? How did we get to eukaryotes? And what does mitochondria have to do with any of this? Well, these bacteria have two membranes, right? So let's draw another little bacteria. Let's draw another little bacteria here. We'll draw it with two membranes. Got our one membrane. Got our inner membrane, like so. All right, we got our two membranes. This will be a gram-positive bacteria. And these make a lot of energy. Okay. All right, now let's say we got another bacteria, but it's a lot bigger. We got this big old bacteria here. Whoa, it's a big bacteria. And maybe one day this bacteria is like, I'm feeling a little hungry. What if I ate a small bacteria? That's the question it asked itself. What if I did that? What if I just engulfed that guy and brought him inside? Copy that boy over. Boom! This boy's making a lot of energy. Well, maybe this bacteria now divides inside of it. Boom! Maybe we get even another one. Maybe we got three. Uh-oh. That boy is broken. Sometimes that happens. Boom. See, what? No. Sometimes this be happening to me. All right. Boom. Okay, now I have three little energy producers in me. This is great. This is wonderful. This big bacteria swallowed some small bacteria and made them make energy for it. It's pretty cool. Bacteria. Something's going on here. What is this? Don't want that. This whole little thing is pretty nifty. This is called endosymbiosis. And this is how we made eukaryotes. So big bacteria decided to make little bacteria its bitch. Whip them little boys into shape and have them make energy for it. So now these little guys be working for the man. They be making ATP for the big boy. It's pretty cool. It's pretty cool indeed. They make ATP for the big boy. And that's what a eukaryotic cell is. Eventually it kind of pinched off some of its membrane. Here, swallowed something else to make a nucleus. If you look at the nucleus, it's actually got two layers around it. So we think that kind of swallowed another cell, and then that became the nucleus. And then every eukaryotic being that is now alive came from this first primitive eukaryotic cell. So like everything from, just plants, fungus, and animals, just like to name a few, all came from this common ancestor. It has a nucleus. These little things here that make ATP energy, these would be called mitochondria or mitochondria. If you're feeling cool, one of my students pronounced that way, I really like it. But mitochondria, and these make ATP. And we'll talk about how all that is, how all that works. But they have two membranes. They actually have pretty nifty. If we go back here and look at this, look at this. Bacteria have circular DNA in the center in a nucleoid. I mean, guess what mitochondria have? I mean, you don't have to be a genius to figure this out at this point. But they got circular DNA. Like that. And it looks like a nucleoid. So not only does your nucleus have DNA, we'll talk about that, but your mitochondria have DNA. And they essentially have bacterial DNA. Your mitochondria, you have tiny little bacteria living inside of you. We've just whipped them into shape and we have them do our bidding. It's pretty cool. All right. There's more that I could talk about here, but I think that's all I want to talk about. Prokaryotes, they come in some different shapes. So this would be a bacillus. This would be a bacillus. Let's talk about the shapes. There's only three. So they got a bacillus, which is the little rod-shaped. So they look like that. They look like little rods or pills. Rods or pills. Then we got coccus or cocci. These are little spheres. And then we got spirillia. Spirilium. That's really all I care. There's also spirochete or spirochete. I don't care that you know the difference. But then we got spiral-shaped bacteria. Something like that. Spirilium. And then bacteria can be either staph or strep. Very quickly. Staph means they form clusters. They form clusters. So a staphylococcus would look something like this. They form little like clusters of grapes. All right, strep means they form long chains. So a streptococcus means that they would form big long chains like this. So if you have strep throat, that's caused by a streptococcus. If you look at a swab of your throat, you'll see bacteria that look just like that. It's pretty cool. We've actually had some students have strep throat, and we do a swab of it and all that stuff. You do that, you'll do that in micro. You'll swab your throat, and if you have strep throat, you'll see that stuff. Okay, that's all I wanted to talk about. Pretty wild stuff here. We have bacteria living inside of every one of our cells. We call it the mitochondria, the powerhouse of our cells. They make our ATP. We'll talk more about how that works in the future. All right, guys. I'll see you in the next one.